Hello, this is Dr. Steve Feldman, Professor of Dermatology at Wake Forest Baptist Medical Center in Winston-Salem, North Carolina. I'll be discussing the publication Key Design Considerations on Comparative Clinical Efficacy Studies for Biosimilars, adalimumab as an example, by Dr. Lai and colleagues. This study report was published in Rheumatic Musculosketal Disease. I selected this article because it provides a review of 9 phase 3 global clinical efficacy studies that highlight key study design considerations of proposed biosimilars. This study uses adalimumab as an example to highlight design elements that may deserve special attention, including therapeutic indications, and for our discussion, plaque psoriasis specifically, target patient population, background therapy, blinding, stratification, transition design, which is a switch from originator to biosimilar product, primary dependent variable, choice of equivalence versus non-inferiority design, section of equivalence margin, and alternative statistical considerations. An equivalence design is thought to be more rigorous, demonstrating biosimilarity of a biosimilar versus the branded product. This review will help clinicians understand regulatory requirements for biosimilar development and design of pivotal clinical trials to demonstrate comparative efficacy and safety for biosimilars versus branded products when considering prescribing biosimilars for rheumatic diseases. Before beginning my analysis, let's hear about some of the study's highlights from the lead author's perspective. What do you feel are the 3 most important highlights or findings of the study? To demonstrate biosimilarity and not efficacy and safety de novo, phase 3 comparative clinical studies for biosimilars have some design features: equivalence or non-inferiority design, switch from the reference product to a biosimilar or vice versa, distinct from those for novel biological products. Key study design elements vary among the 9 phase 3 studies for 8 adalimumab biosimilars in terms of the disease indications, target patient population, background therapy, disease activity, transition design, primary endpoint, equivalence margins, etc. While each biosimilar is compared with the reference product in these studies, due to the differences in study design, it may be difficult to compare the different biosimilars with each other. Even if the reference product has been approved for multiple indications, pivotal phase 3 studies to demonstrate comparative efficacy and safety for biosimilars are usually carried out in one disease indication and extrapolated to other indications. For example, out of the 8 adalimumab biosimilars that were or are evaluated in phase 3 comparative studies with the reference product, 4 were evaluated only in rheumatoid arthritis, 3 only in plaque psoriasis, and 1 in both rheumatoid arthritis and psoriasis. What impact do you think this study will have on the utilization of proposed adalimumab biosimilars in patients with plaque psoriasis? By understanding the different phase 3 study design approaches taken with 8 adalimumab biosimilars either approved or in development, the study's findings will hopefully, 1, help clinicians evaluating the comparative efficacy and safety data for the adalimumab biosimilars, and 2, facilitate their discussions with patients on switching to or initiating an adalimumab biosimilar for the treatment of plaque psoriasis. Here are the methods of this study. A psoriasis 2013 phase 3 clinical trial with 448 subjects for GP2017, a biosimilar for adalimumab, permitted enrollment of patients who received prior biologic therapies. The primary endpoint was a 75% reduction in the psoriasis area and severity index. We call that a PASI 75 at week 16 in patients with plaque psoriasis. Psoriasis studies indicated that treatment assignment, weight, and age are the most influential factors for mean % change in PASI score at week 16. Efficacy is decreased mainly in patients with body weight greater than 90 kilograms or with body mass index, BMI, greater than or equal to 30 kilograms per meter squared. The most significant decrease in efficacy occurs in patients with body weight greater than 140 kilograms. Data show that it might be worthwhile to consider weight or BMI as a stratification factor with the biosimilar adalimumab, or to exclude extremely obese patients because obesity tends to be more frequent in patients with psoriasis. Of the 5 indications approved for adalimumab, the greatest placebo-adjusted response rate was found in psoriasis, 61 to 64%. Psoriasis may represent a more sensitive disease model to detect any potential difference in immunogenicity of the biosimilar versus branded adalimumab. Ongoing consultation during the development of a biosimilar product is necessary to make adjustments as necessary based on preclinical and clinical data because the totality of evidence is used for regulatory approval. Here are my thoughts and analysis of this study. To get a biosimilar approved, you must show that it is similar to the innovator biologic. You can do all kinds of preclinical tests to show the products are similar, but to alleviate any residual uncertainty that the drugs would perform similarly, a single clinical trial can be done. That trial should be done with patients who have the condition that would be most sensitive for identifying difference between products. This study makes the case that for tumor necrosis factor inhibitors, psoriasis would be the best condition to study. Psoriasis is easily measured, and because psoriasis is often treated with the biologic in isolation, as opposed to rheumatoid arthritis or psoriatic arthritis, in which concomitant methotrexate is often used, there should be good sensitivity for detecting immunogenicity. The results of this study won't have any immediate impact as we don't have an adalimumab biosimilar on the market in the United States. In the future, this study should give us great confidence that adalimumab biosimilars can have similar actions as the innovator products. I think many people believe a lot of questions remain unanswered. Here are a few. Will doctors be comfortable prescribing a biosimilar that is not identical to the original drug? Will we accept patients being switched from originator to biosimilar without our knowledge? Will we be satisfied if biosimilars are tested in patients with rheumatoid arthritis and not psoriasis? Will we be comfortable with potential differences in immunogenicity, safety, and efficacy? Will we be comfortable with the biosimilar using the same generic name that the originator drug uses? I think these questions are easily answered knowing that different batches of the innovator product vary too. So, will doctors be comfortable prescribing a biosimilar that's not identical to the original drug? We have been comfortable giving patients different batches of the innovator, and they're not identical. Will we accept patients being switched from the originator to biosimilar without our knowledge? Patients have been switching between different batches of the innovator without our knowledge. Will we be satisfied if biosimilars are tested in patients with rheumatoid arthritis and not psoriasis? The current batches of innovator have not been compared to the original batch in any clinical trial. So if we are comfortable with no trial at all, we ought to be comfortable with a rheumatoid arthritis trial. Will we be comfortable with potential differences in immunogenicity, safety, and efficacy? We have been comfortable with potential differences in immunogenicity, safety, and efficacy between different batches of the innovator, and we'll have more data on biosimilars showing that there aren't differences in immunogenicity, safety, and efficacy. Finally, will we be comfortable with the biosimilar using the generic name that the originator drug uses? We've been comfortable with different batches of the innovator using the same generic name. I would be comfortable if well-tested biosimilars use that generic name too.